Good afternoon. My name is Sung-Wook Nam in the Department of Molecular Medicine. Thank you for having me to the last talk. And I'm very happy to present this Hamamatsu symposium. So my talk is about image tracing of inflammatory intestinal organoid by using computational clearing, which is a kind of image processing algorithm. So I will introduce this one. So let me briefly introduce my laboratory. So my expertise is engineering and technology. So in our laboratory, we develop intestinal organoid growth, and we would like to apply this system to device or chip, intestine on a chip and vertebrate chip. Eventually, we just pursuing the drug screening. And also, we developing anaerobic chamber, so microscopic imaging technology, and prototype device development of device apparatus. So among them, today I'm talking about imaging technology. These days, there are many automation of image analysis. Normally, it is consists of a normal process of normalization, registration, and computation and interpretation by AI and machine learning. Among many automated image analysis, we adopt computational clearing method. So here is the wide field image of conventional fluorescence microscopy, and real world is that it's where background noise is many and very hazy fluorescence signal. So biomedical researchers normally use confocal microscopy, which shows very nice images. However, confocal microscopy is a bit expensive. So our team, by adopting this computational clearing algorithm, image processing algorithm, we want to obtain confocal level images, fluorescence image, because this is highly beneficial for further image segmentation analysis and mesh generation to extract physical, morphological parameters. I would say this one, computation clearing CC is good for image improvement. And the idea, our hypothesis is that during the image process from wild type image to CC image, there should be some differential signal, the difference signal between wild field WF and CC. So we want to actively utilize the signal difference, and finally we want to apply that idea to phenotyping or diagnostic application between disease model, inflammatory model, and control models. To do that, our lab established long-term culturing of mouse intestinal organoid. We isolated the crypt of mouse intestine and embedded in matrigel, which serve as extracellular matrix, and we cultured EGF, login, R-spondin complete medium to grow mouse intestinal organoid. We use transgenic mouse in which LGR5 is the marker of intestinal adult stem cell. This LGR5 is tagged by EGFP, green fluorescence protein, and more than 20 days culturing, we could obtain almost 1 millimeter size. This is 24-well dish, and 1 millimeter size mini organ we could culture. And we monitored the organoid area for each passage. And after growing, we identified that the green fluorescence, actually intestinal adult stem cell is flanked by Pannis cell, so it's repeated structure. So we could image such a fluorescence signal. This is movie. I'm sorry. Okay. Okay. Let me show you this real-time image for live dead imaging. Almost 4 days we monitored the growth of organ. Actually, this is swelling with the thin epithelium while collapsing that the epithelium becomes thicker. We believe that this is one evidence of development of intestine folding structure based on this real-time movie. So now we need some disease model. So we treated dextrans sodium sulfate, which is widely used for to induce to in vitro or in vivo model of inflammation. So after growing 7 or 10 days of mouse intestinal organoid, we treated it with DSS, dextrans sulfate sodium. Here, a surface group attack some mouse intestinal organoid to induce inflammation. So this longitudinal study for days and then the titration of the DSS concentration. So control sample has normally grow while the DSS treated samples induced some collapse of epithelium shown in this picture. And after culturing, we performed a live dead assay shows dose-dependent manner. So control samples as previously image movie shows that this intestine organ is very fast and rapid turnover. So apoptotic cells are accumulated inside the lumen. So blue color is live cell and red color is dead cell. So this apoptotic dead cell is accumulated inside the epithelium, tight epithelium, while inflammatory organoid, in the case of inflammatory organoid, the dead cell is spilled over, so distributed outside the organoid, which shows the is a one proof of induction of inflammation. We checked the gene expression of inflammatory mouse organoid. So firstly, intact organoid has this green color intestine stem cell existing here, but inflammatory inflammation is induced. This green color is off, removed, and also we checked the intestinal stem cell marker. So among them, enterocyte, goblet, and endocrine, the gene expression level is almost maintained. However, stem cell consisting cells like a stem cell and Paneth cell is decreased as the treatment of DSS. And we checked the mRNA sequencing that here not shown very clearly, but OLFM4, which is the marker of another marker of the stem cell region, is significantly down-regulated. And also we checked the signal. So inflammatory related signals like interleukin JAK and TNF alpha, NF-kappa B is unregulated, while cell survivor or stem cell signal like PI3K-AKT and Wnt beta-catenin is downregulated. So based on this model, we think that this stem cell niche region consisting of stem cell niche region is damaged by DSS drug, while this villus region is still alive, and also gene markers can be shows the same behavior. So what is the computational clearing? So to explore this one, we adopt very thin bottoms of slide glass, and then we could image by using 63x oil immersion lens with the numerical aperture about 1.4, and then we 3D-dimensional G-stack image with 2 micrometer slicing, and after that we staining in DAPI and phalloidine, which shows very nice 3D-dimensional structure. So one thing is that in phalloidine image, actually this intestinal epithelium has a polarity, so apical and basolateral. So F18 is normally expressed in the apical side. So here, apical in inner region is strongly F18 is expressed, which means that this our organoid is apical in structure. And based on this 3D-dimensional structure, we performed the cell segmentation, followed by mesh generation. We could estimate the volume or physical parameters like the surface area of these fluorescence signals. So finally, we would like to explore our hypothesis, kind of image tracing. This image tracing means that tracing from wild field to computational clearing. So CC minus WF. Actually, this is our suggestion, hypothesis. Upper one is intact organoid. Upper one is intact organoid. And here is the intact organoid. This is inflammatory organoid here. We tested 10 to 20 mouse organoid. In intact one, the following signal is very sharply identified at the inner lumen regions, while DSS treated one shows very broad, implying the destruction of the epithelial cell. And now we apply the wide computational clearing by comparing between wide field and computational clearing. Wide field is not shown in this screen, but very broad background noise is still here, but that is almost completely removed by CC processing. And also DSS treated one also shows same tendency. So based on these two images, for wide field image, we try to phenotyping between intact and inflammatory organoid. And also by using CC image, we try to do that. So in more sense, it is possible to distinguish two different phenotyping. However, our assumption is that it will be more enhanced if we tested the differential signal from CC minus wide field. So actually, this plot is the circularity and area, organoid circularity and area, and each point implies each one feature of a mouse intestinal organoid. So by comparing wide field and computational clearly, we could calculate the vector like a shift of circularity parameters and a shift of area that is plotted here. So in control samples, the shift is not that high, almost distributed at 0.0. However, double-stranded DSS treated one, and some shifting while image treatment. And based on, we could do phenotyping diagnostic application to distinguish two different phenotypes. So the question is, why do we have to CC minus W active? So we think that in current camera technology, camera detection, very sensitive to enhance the non-specific binding, just enhancing. So it's like image fabrication. So that's the disadvantage. To overcome that, we think that such a CC and minus Y, the differential signal will be mitigated that risk. That is one of the reasons, and also provide more consistent and reliable diagnostics. And finally, we need to compare. We need to check the comparison between confocal microscopy image. So yes, confocal microscopy is good, and also our computational clearing based on wide field image. We tested the DAPI and FFT and ZO1 staining. ZO1 is marker of the tight junction. So by using this CC method, we clearly identify the ZO1 expression, tight junction formation, and even more further analysis, image of segmentation and mesh generation, we could extract the physical parameters like a volume and surface area. So conclusion is that we adopted computational clearing, which is very, very good for obtaining high quality image, and also we come up with CC minus wide field theory. We hope that this technology can be used for diagnostic application to discriminate injured state and intact state. So we are now testing about we see the possibility of this theory demonstration. So I would like to acknowledge my laboratory member, Dr. John, and thanks for your attention, and I'm happy to receive your question.
Thank you, Dr. Nam. So any questions? Yes, here.